Hey guys, how you doing? So welcome to the last lesson of chapter 10, looking at asexual reproduction. So in this lesson, we're going to look specifically at how asexual reproduction is used in our modern day, and then also the issue of cloning. Okay, let's get into it. Okay, so asexual reproduction and its uses. Now, in agriculture and horticulture, this is probably where asexual reproduction is used the most. Plants are easy to reproduce asexually. That's the key thing there. Like you saw with an example of bananas, you can do vegetative propagation and you can multiply their numbers quite easily rather than waiting for the plant to get pollinated, produce flower, fruit, and then seeds, and the seeds drop and grow into new plants. Yeah, and so you've got this kind of trade-off of you're losing genetic variation, which doesn't quite matter if the quality of whatever you're trying to grow is pretty much all the same, but you're gaining that speed. Yeah, you can create it en masse, and economically it's very beneficial. You only need one part of the plant to produce many copies, and so there's an advantage of asexual reproduction in that you don't lose, you have this kind of backup where you have one segment of the plant, and things like that. You can then reproduce and repopulate a particular area. Yeah, tissue culture. Tissue culture is basically when you grow plants or new plants inside a gel rather than inside the ground, and so you can keep reserves of plants in tissue culture, which is a safe environment where the environmental factors are controlled, the day length, the sunlight exposure, and things like that, to make sure that the plant survives. Now, what do I mean by that? I'll get to hormones in a second. So this is actually a tissue culture of a particular flower, yeah, and so they put a portion of it into a jar, and then it's grown in this kind of agar gel, and so the plant stays alive, and so long as it receives the nutrients it needs, this is a backup of whatever that flower is, right? And so what you can do is you can take the tissue culture, move it, transport it to different places, and then you can isolate it, grow it in the ground, and you have and propagate it to produce fields of it. Yeah, and so what you've got is that you've got large masses of flowers of the particular color you want, the particular strain you want, and therefore you can economically take advantage of that and sell it and things like that. Yeah, same can be done for fruits and vegetables and things like that. The other advantage of using asexual reproduction is that if you have something like a tissue culture, you can then isolate it, you can add hormones, and you can grow a particular trait or a particular quality of the plant or the vegetable or whatever, and then you can propagate it once again. And so it's a very quick way to multiply the effects of the hormone, yeah, and in economic terms that's quite advantageous. Yeah. Now, the other part I want to talk about is cloning. So in mammals, this is sort of where the big debate sort of happens, is that up until now we've looked at asexual reproduction in many animals, but never a mammal. The reason is because mammals naturally only reproduce through asexual reproduction. That is, you need the sperm and the egg to form a zygote. Yeah, the zygote being that first cell in a new organism. Yeah, and that first cell is only produced through fertilization. When the sperm and the egg combine, it forms that new individual, and that new individual from there undergoes mitosis and grows into that organism. Okay, and so cloning in mammals is trying to bypass that in effect. Yeah, so what you've got is that you're trying to skip this combining of egg and sperm, and you're trying to produce a new individual that has the same DNA as the parent without that combining of genetic material. Yeah, and so that's what cloning is attempting to do. Okay, and there's a few different ways that we can do it, and there's a few very interesting methods that they've done back in the past couple of decades. The first one is called embryo splitting. Now, this is kind of like cloning, but also kind of not like cloning. Yeah, embryo splitting is when you've got a large embryo and you actually divide it into smaller embryos. Yeah, it's basically the same process of replicating identical twins, except you're using it in kind of livestock and things like that. Okay, so each embryo that's been split up is planted into a surrogate female, so a female that wasn't the original mother, and then it's grown into a full-grown offspring. Okay, so for cows, dairy cows and things like that, the advantage is that you can impregnate multiple females at once using the same batch of embryos. Yeah, so if you've got a a male bull, you take the sperm from the male bull, you take the a female cow, and you artificially inseminate the cow with sperm from the bull. And so what happens is the female gets pregnant, produces an embryo, and then what you do is once the embryo gets to a large enough size, not too big that you cannot split it, but large enough size that you can then be split it into smaller clumps of embryos. Yeah, and so here you've got many more times the number of embryos. And remember, these embryos are all identical to each other because they've already combined, formed the zygote, and the zygote has gone through mitosis to form new embryos, and so that gets split up into many more clumps, and you can impregnate foster mothers or foster cows with more of these embryos to get them all pregnant at once, producing many more offspring. This is a very straightforward method because you're still cloning the embryos. However, it's worth noting that you still need the sperm from the male in order to produce the embryo in the first place. Yeah, and so that's one of the kind of the key struggles with this embryo splitting method. Yeah, scientists have worked really hard to sort of try and get around some of these methods, and one of the methods that they've actually used and developed over the years is what we call somatic cell nuclear transfer. Yeah, now this sounds like a bit of a mouthful, but let me break it up for you. Yeah, the somatic cell is a non-sex cell. It's any cell in the body that is not the sperm or the egg. And so nuclear refers to the nucleus, which is the part that you're trying to transfer because that's the part that has the DNA. Yeah? And so somatic cell nuclear transfer is trying to transfer the nucleus of a somatic cell into an egg. Yeah? And I'll explain what that looks like in a second. So you're cloning a normal cell, sorry, you're using a normal cell in the body and you're moving its nucleus into an unfertilized egg to produce a zygote. Yeah, and so here is Megan and Morag, which are two Welsh ewes which were cloned in 1995. Yeah, and the way that they were cloned was using this method of somatic cell nuclear transfer. Yeah, what they did was they got the nucleus removed from an unfertilized egg. So there's your unfertilized egg, and the nucleus was actually taken out. So that egg is now a bit of a blank cell, has no DNA, but it has all the necessary organelles to produce a new organism, yeah, except for the nucleus. They took some donor embryos from another ewe, and then they took the embryos out, and what they did was they put the embryos next to this unfertilized egg, and then they put an electric pulse in it. And what that electric pulse does is it fuses the embryo into the fertilized egg. The embryo contains the DNA, right? And so by doing that, it fuses the DNA or the nucleus of the donor cell into the unfertilized egg, forming a new type of cell. Yeah, one now fertilized egg with the DNA of the embryo. Yeah. So what that means is that this egg belongs to the individual here, but it has the DNA of this individual here. And so you've got a cloned version of this individual with the DNA, and that egg can then be implanted into a surrogate mother and grown into a full offspring. Yeah. Now, one thing to note is that the scientists that sort of did this particular experiment did it using existing embryos. Yeah, and so the embryo here already has the DNA and it's already formed, and then they took the embryo, fused it with another cell to try and get the DNA to fuse. Right? You can do this. You don't have to do it with a embryonic cell. You can do it with a normal cell. Yeah, which is what they sort of worked on over the years. Yeah. If you don't take an embryo, you can take a regular body cell. So same concept. If I was to show you, let's say you've got sheep A, and what you do is you take a body cell, a muscle cell, or a skin cell, or whatever segment of a regular body cell from the sheep. You take the DNA out of this, right, from sheep A, and then you take an egg from sheep B, an unfertilized egg. You do the same thing. You remove the DNA. And so that it's this kind of blank, empty cell. You take the DNA of one cell and you fuse it with the shell of another cell, So you now have a DNA from sheep A fused with the egg from sheep B, yeah? And so that is a fertilized egg, yeah? Through artificial means, obviously. But then that egg will now be able to divide and form an embryo, a cluster of cells, and then once it grows large enough, you put that clump of cells into a different sheep altogether, and then it will produce a lamb that has the same DNA as sheep A, yeah, because that's where the DNA comes from to produce that lamb, yeah. Now, a famous example, Dolly the sheep, was cloned in 1996, and she was the first successful clone using this method of somatic cell nuclear transfer, yeah. So she was the offspring of an artificially cloned embryo, and then she grew into not only a lamb that was, into adulthood, but she was able to then be a fertile lamb and produce offspring of her own. So that's Dolly the sheep there, and that's actually her offspring, yeah? And so this is kind of the breakthrough moment in science around the concept of cloning, because this was for the first time that you didn't need sperm in order to reproduce an entire mammal, yeah? Mammal being the key part here, because they can, you can do it with lizards and insects, but to do it with a mammal was a real breakthrough moment, okay? So why was she so famous? Here is an article that Ian Wilmot wrote about Dolly. So Dolly seems a very ordinary sheep, yet as all the world acknowledged, she might reasonably claim to be the most extraordinary creature to be born. Dolly has one startling attribute that is forever unassailable. She was the first animal of any kind to be created from a cultured, differentiated cell taken from an adult. Thus, she confutes once and for all the notion, virtual dogma for 100 years, that once cells are committed to the task of adulthood, they cannot be totipotent again. Yeah, and so that's a bit of a breakthrough, because the idea that a cell can be totipotent once more was proven by Dolly the sheep. Okay. Now, there's obviously, kind of the upside of this breakthrough in science, but there's also a few problems with this, okay? And so let's take a look at some of the issues there. So Dolly actually took 277 attempts, which means 277 unfertilized eggs, 277 samples of body cells, 277 embryos that formed and then died as a result before Dolly came along. And so many embryos were fused together with normal DNA and didn't make it, didn't survive, died as a result. And so you kind of have to question, well, that number of attempts and would equal means that number of potential life that was killed as a result. In general, clones have developmental issues, yeah? About 1 in 6 are healthy, and many of them die during that early stages of development. So they succeed in fusing the egg with the DNA, but then it grows into the embryo and then it just dies along the way, yeah? They also notice that even after the clones are born, they generally exhibit challenges in their life. All clones generally have a shorter lifespan than the individuals, and later on when you look at the structure of chromosomes, you'll actually understand why that is. And they also underwent lots of health issues as well. Now, aside from the actual, the physical and scientific problems with this, there's also ethical issues with cloning, right? And so you have to ask yourself the question, in terms of applications, if we can clone sheep from other sheep, then are we able to then clone parts of whole organisms for products? Yeah, kind of the thought process there is that if we can do it with sheep, then we can then clone parts of people, yeah, organs that are important for organ transplants. You can have, rather than relying on organ donors, you can now replicate the organ of an individual through a clone and save a person's life. And so these applications, kind of open this new door to all these possibilities. But once again, it presents all these ethical questions of, even though we can, should we? Yeah. Regrowth of limbs and other vital organs, some real breakthrough in medicine and science there, but then also thinking of, the life that you've created and then culling that life in order to harvest those organs and limbs and so forth, being an ethical question of debate. And so there's opposition for that. You are interfering with nature. Essentially, it's an artificial process. Humans have always interfered with nature, but this kind of seems like one extra step away. And then also the idea of playing God. You know, as humans, are we supposed to create new life through artificial means rather than through natural means? And are we playing God in that process? So those key questions are worth thinking about in terms of the possibilities, but also the ethical issues surrounding cloning of animals. Yeah, as you guys are learning this, I really want to challenge you guys to think about these issues deeply, and the idea because of cloning, because if you ever go into this field, into this area, and these breakthroughs in science happen, it's worth thinking about whether or not people are meant to be doing these things or not. Yeah. So hopefully you guys learned something there. We'll talk about more in class, but that's it for chapter 10. Hope you guys enjoy. See ya.